Hello learners, welcome to online course on functional foods and nutraceuticals. I am Dr. Rekha Sharma, Associate Professor, UGC Human Resource Development Center, Rashtrand Tukaroji Maharaj, Nagpur University, Nagpur. Learners, under the non-nutrient specific effect of nutrients, we shall study one of the most important bioactive nutrients, alpha linolenic acid, ALA, commonly referred to as omega 3 fatty acid or N3 fatty acid. Omega 3 fatty acid is an essential fatty acid as it cannot be synthesized in the body and must be supplemented in the diet. On consumption, it is the precursor of long chain fatty acids eicosapentaenoic acid EPA and docosahexaenoic acid DHA. Omega 3 fatty acids are important structural components of cell membrane, serve as precursors to bioactive lipid mediators or provide a source of energy. Omega 3 fatty acids exert some of the powerful health benefits for human body and brain and are emerging as some of the most widely used beneficial compounds in human health. This module will brief you on alpha linolenic acid, its chemistry, classification, dietary sources, metabolism and bioavailability and its physiological effects and health benefits and possible food applications. Now, let us study the definition and chemistry. We know that fats are made up of fatty acids and glycerol. Fatty acids are carbon chains of varied lengths ranging between 4 to 22 carbon atoms. These fatty acids are classified according to carbon chain length, according to degree of saturation, according to position and configuration of double bonds in chain length, and according to the degree of saturation they are classified as saturated and unsaturated fatty acids. Saturated fatty acids have all single bonds, whereas in unsaturated fatty acids carbon atom is connected with adjacent carbon atom with a double bond. Now, from the nutritional point of view, there are three essential unsaturated fatty acids, namely linoleic, linolenic and arachidonic acids, which have 2, 3 and 4 double bonds respectively. They are said to be essential since they cannot be synthesized by the body and need to be taken through diet. Now, let us see the positions of these double bonds in these fatty acids which decide their names or nomenclature. Fatty acids are made up of carbon chains, one end of which has carboxyl group known as alpha end and the another has methyl end, that is CH3 or known as omega end. The position of double bond is counted from the methyl group of the chain and denoted as omega or also noted as N. Linoleic acid has a chain of 18 carbon atoms and has 2 double bonds, and the position of first double bond from the methyl end is 6th carbon atom. Hence, it is known as omega 6 fatty acid. Linolenic acid has 18 carbon atoms chain, but has 3 double bonds, and the position of the first double bond from methyl end is 3rd position. Hence, it is called omega 3 fatty acid. Arachidonic acids have 4 double bonds and the position of the first double bond from the methyl end is 6th position, hence called as omega 6 fatty acid. Let us now study the concept of cis and trans fatty acids. The arrangement of hydrogen across the carbon atoms is referred to as configuration. On the basis of configuration, fatty acids are categorized as cis and trans fatty acids. In cis arrangements, the hydrogen atoms at the 2 carbons linked by double bond are on the same side. In the trans arrangements, the carbon atoms in the chains are on the opposite sides of the double bond. in a fatty acid with cis form, the acyl chain will be bent at an angle. This gives some freedom of movement to the fatty acid. In trans fatty acids, the acyl chain is more or less straight and is rigid. Fatty acids in vegetable oils are naturally in cis form that can be converted into trans during process of fats and oils. Now, after studying the basic concept of nomenclature, now let us study in detail about omega 3 fatty acids. Now, after studying the basic concept of omega, let us now study about omega 3 fatty acids. There are 3 types of omega 3 fatty acids whose first double bond from the methyl end is placed in the third carbon atom. These 3 types are alpha linolenic acid, that is ALA, eicosapentaenoic acid, that is EPA, and docosahexaenoic acid, that is DHA. Now, let us study what are these alpha linolenic acid, eicosapentaenoic acid and docosahexaenoic acid. Alpha linolenic acid is essential because it is the parent compound of long chain fatty acids, that is EPA and DHA. The body partially converts ALA to EPA and DHA, but this is not enough to meet the requirement of EPA. Eicosapentaenoic acid, also known as EPA. EPA is converted into several eicosanoids that have anti-atherogenic, anti-thrombotic, anti-inflammatory and immunosuppressive effects. It is also converted into resolvins, that is lipid derived inflammatory modulator, that have anti-inflammatory effect. Thus, it has shown beneficial effects on blood clotting, platelet aggregation, triglyceride level, immune functions and inflammatory responses. It competes with omega 6 fatty acids and produces the more beneficial eicosanoids. EPA is a precursor of DHA. Now, docosahexaenoic acid. Docosahexaenoic acid is vital component of phospholipids present in the cell membrane of the body cells, especially of the retina, myelin in brain and other structures, and is greatly influences the membrane function in these tissues. It is present in grey matter of the brain and influences thinking, concentration, cognition, memory, feeling and other brain functions, particularly during fetal stage, infancy and childhood. However, in later adulthood, it affects several brain activities because it is a messenger between brain cells. Its deficiency can cause neuronal disorders. Following consumption, most of the alpha linolenic acid is catabolized via beta oxidation for energy generation, and a small proportion of it undergoes conversion to produce another 2 potent members of omega 3 PUFA family, that is EPA and DHA. Now, let us study the sources of omega 3 fatty acids. Rich sources of alpha linolenic acid are nuts and seeds and their oils. Flax seeds, walnuts and their oils are among richest dietary sources of omega 3 fatty acids. Canola oil is also an excellent source of omega 3 fatty acid. The other omega 3 fatty acids EPA and DHA are both found in fatty fish and shellfish. Cold water fishes like salmon, herring, mackerel, tuna and sardines are the best sources of omega 3 fatty acids. Animal origin oils like cod liver oil, fish oil, krill oil and vegetable oils like canola, safflower and soybean are also sources of this fatty acid. Foods like eggs and beverages can be fortified with omega 3 fatty acid. Oily fish are the major dietary sources of EPA, DHA and omega 3 fatty acids. Eggs produced by hens fed a diet of greens and insects contain higher levels of omega 3 fatty acids. Fish oils are added to the diets of hens to increase the omega 3 fatty acid concentration in eggs. Enriched eggs are available in the developed and developing nations. Omega 3 fatty acids are formed in the chloroplasts of green leaves and algae. While seaweeds and algae are the sources of omega 3 fatty acids present in the fish, grass is the source of omega 3 fatty acids present in grass fed animals. Flaxseed are a good source of alpha linolenic acid, which also provides proteins, minerals, fiber and plant lignans. Chia seeds and walnuts also contain significant amounts of omega 3 fatty acids. Now, let us study the effects of processing on omega 3 fatty acids. When omega 3 fatty acids are overheated, the fatty acid composition begins to change and breaks down, which means less omega 3 post processing, which involve overheating. The higher temperatures during cooking potentially degrades the EPA and DHA through oxidation. Due to commercial processing, the omega 3 content of oils can change as fatty acids get degraded. The oils are heated to high temperatures in order to refine them and are further heated during cooking. Deodorization is a step in the process of oil refining, wherein the oil is heated to a temperature of around about 200 degrees Celsius. At temperatures as high as this, the basic structure of the oil can change into a form which can be harmful for the body. The process of change in the structure of fatty acid is known as isomerization. While selecting cooking methods for fish rich in omega 3 fatty acids also needs some caution. Low fat cooking techniques such as broiling, steaming, poaching, baking, stir frying, sauteing and grilling are recommended over deep frying, and overcooking should be totally avoided. Exposure to high heat causes the formation of some trans fatty acids. Foods containing omega 3 fatty acids cooked at higher temperatures for long time has no longer original health benefits, since most of the omega 3 fatty acids have been destroyed. Such preparations have no beneficial effect on the body and should be avoided with olive oil, coconut oil, avocado oil and grass fed organic butter. Most scientific studies observed the effect of processing and cooking on omega 3 fatty acids by using variety of N3 rich fish species. A study conducted showed that boiling, steaming, microwave baking and grilling did not affect the fatty acid composition. Pan frying and deep frying increased the general polyunsaturated fatty acid content and decreased the omega 3 and omega 6 ratios, especially for deep frying cooking methods. To summarize, it is best to consume boiled, baked or steamed fish that help in preservation of the omega 3 fatty acids. Omega 3 fats found in other foods like flax seeds, kale or spinach may be preserved by consuming them fresh, which may otherwise undergo oxidative changes and turn rancid.